All right. It's my pleasure to introduce Renata. She is currently a PhD candidate in animal science at the Federal University in Brazil. And last year, she had the opportunity to do an externship at the Aquatic Animal Health Lab at the University of California, Davis. She earned her master's degree in veterinary sciences in 2019 and her veterinary medicine degree in 2017. Her research interests primarily focus on aquatic animal health, epidemiology of infectious disease, and disease prophylaxis. Take it away, Renata.
Thank you. So I'm Renata, I'm from Brazil, and today I'm presenting some of the results from the time that I was at Dr. Soto's lab. And we are talking about the association between genetic diversity and virulence of Edwardsiella species in tilapia from Brazil. So Brazil is located here in South America. And when most people talk about Brazil, we think about soccer, Pelé, Ronaldo Fenômeno, Marta, and also beautiful beaches and carnival. But actually, Brazil is also an important player in the aquaculture scenario. In the last 9 years, fish farming in Brazil increased approximately 50%, and the Nile tilapia is the most produced fish in the country, representing approximately 64% of all fish produced here. And Brazil nowadays figures as the fourth largest Nile tilapia world producer. Here in the country, tilapia is mostly produced in the south and in the southeast of Brazil. And it's interesting for you to know that the Brazilian legislation doesn't allow us to have any tilapia farms in the north, where we have the Amazon forest, because this is an exotic species and it could be prejudicial to our native species if it got into the Amazon ecosystem. We also produce many native species that represent 31% of all fish produced here in Brazil. And here's the tambaqui, which is the most produced native fish in Brazil. And these fish are mostly cultured here in the north of the country, mostly because of the climate, because here we have warmer temperatures that these fish prefer. And when we increase aquaculture, we also increase fish diseases, not only in Brazil, but worldwide. And in Brazil, the main fish pathogens are Streptococcus species, Lactococcus, Aeromonas, Edwardsiella, Francisella orientalis, columnaris causing agents, and the ISKNV. So here today, we are focusing on the Edwardsiellas, and there are 4 species in the genus that are pathogenic to fish: Edwardsiella tarda, E. ictaluri, E. angularum, and E. piscicidum. Fish infected by all these Edwardsiella present non-specific clinical signs, and as Kim already presented, it is very common for us to see this multifocal petechia in the fish skin, and fish can also have septicemia, this is when the Edwardsiellas reach the fish brain. It is also very common for us to see these multifocal white nodules in the fish spleen and also in the kidney. And in the histology, we can see that these white nodules reflect the presence of granulomas in the organs. So what's the scenario of the Edwardsiellas in Brazil? Nowadays, we don't have commercial vaccines against Edwardsiella available in Brazil, and therefore antibiotic therapy is the main strategy for treatment and control of the infection. And actually, little is known on the diversity of members in this genus. Until very recently, we thought we only had Edwardsiella tarda here in causing disease in fish in Brazil, but very recent study proved that we also have Edwardsiella angularum here. So the main aim of our study was to determine the taxonomic classification and the genetic diversity of Edwardsiella species affecting Niotilapia in Brazil, and to understand the bacterial virulence. And for that, we selected 40 isolates of Edwardsiella from diseased Niotilapia farmed in different regions of our country that were collected between 2015 and 2022. So we had 7 isolates from the Northeast, 25 from the Southeast, 5 from the Central West, and 3 from the South. We didn't have any here from the North because we are working with isolates from tilapia. And as I mentioned, we don't have tilapia here in this region. So as I mentioned, we collected these isolates from tilapia that were sent to our lab, and we isolated the Edwardsiellas in Shibboleth agar. Following this, we extracted the genomic DNA and sequenced it with Oxford Nanopore technology, and then we assembled the genomes. For the taxonomic classification of our isolates, we did a phylogenetic analysis based on the sequence of the DNA J gene. And to analyze the genetic diversity, we used the MLST. To assess the presence of bacterial virulence genes in our isolates genomes, we used the BVBRC platform. And after we analyzed the presence of these virulence genes, we did a in vitro assay to understand the virulence of our isolates to the. And for this, we used ONB cells that are cells derived from Tilapia mosambica brain. This tilapia is cultured worldwide, and the brain is an important target organ for the Edwardsiellas. Here in this video, we can see some tilapia infected with Edwardsiella, presenting a very characteristic clinical sign, which is the diuretic swimming and loss of balance that happens when the Edwardsiella reaches the fish brain. And for that, we generated bacterial suspensions that we added to 24 well plates with our ONB cells to reach a MOI of 1 to 10. Then we centrifuged the plate and incubated it. Following, we added 50 micrograms per ml of gentamicin and incubated the plates and washed it twice, and added 0.25 micrograms per ml of gentamicin for our E. tarda isolates and 0.5 micrograms per ml for our E. angularum and E. piscicida isolates. And then we incubated our plates for 24 hours or 48 hours, 24 for our E. tarda and 48 hours for E. angularum and E. piscicida. After this incubation period, we got 50 microliters of the supernatant and transferred it to a 96 well plate in duplicates and added 50 microliters of a reagent from a commercial kit and let it incubate. During this incubation, the reagent reacted with the lactate dehydrogenase, which is an enzyme, cytosolic enzyme that is released by lysed cells. And after this incubation, we added a stop solution and read the absorbance in 419 nanometers to quantify the product of this reaction. So here's our phylogenetic tree of our isolates. We used some Edwardsiella strains that had their taxonomic classification already confirmed. And with this, we knew that we had detected 19 E. angularum isolates, 1 piscicida, and 20 tardas. We also did the phylogenetic analysis based on the MLST concatenated alleles, and we had the same results as the DNA J gene. Following this, We did a evolutionary relationship analysis based on the MLST scheme using the eBurst algorithm, using all STs previously characterized and now our new STs from our studies. And I would like to call your attention for our Edwardsia latarda STs here in gray. I'd like to show you that they are very, very genetically diverse. Following this, we did a phylogenetic analysis based on the MLST concatenated alleles for each of our species. And here are our Edwardsia isolates. And we see that we have 3 main genetic lineages, 1, 2, and 3. And from the genetic lineages, we selected some representative isolates for our next analysis. We did the same thing for Edwardsia angularum, and we had 3 genetic lineages that, as expected, matched the sequence types that we had identified through the MLST analysis. We also selected some representative isolates. For our Edwardsia lepisicida isolate, since we only had 1 isolate and we have more data in the MLSA database, we used the MLSA to analyze the genetic diversity of our isolate. And it was grouped here in clade number 6, where we have another isolate of Edwardsia lepisicida isolated from tilapia. So talking about the virulence genes, first about our Edwardsia latarda isolates. None of our Edwardsia latarda had any genes of type 1, 3, and 6 secretion system. And 1 isolate from cluster number 3 had genes from type 4 secretion system, which hadn't been reported in the literature so far. All our isolates had genes for pili, adenosines, emolazines, superoxidase mutase, and chondroitinase. And some of our isolates had genes for fimbria and catalase. As for E. angularum and Epicicida, all our isolates had genes for type 1, 3, and 6 secretion systems, and this has already been reported in the literature, and none of them had type 4 secretion system genes. All of them had genes for pili, emolazines, catalase, superoxidase mutase, and chondroitinase. Our Epicicida had genes for adenosines, and most of our isolates had genes for fimbria. And talking about our in vitro assays, for the E-tarda isolates, our genetic cluster 2 was the most virulent for the OMB cells, specifically the isolate ED 2716. And for the E. angularum isolates, ST32 was the most virulent for the cells, followed by ST28. And I would like to call your attention that this ST32 that was the most virulent was also the most prevalent. So most of our isolates belong to this genetic lineage. And since we only had 1 Edwardsia lepisicida isolate, we compared it to another Epicicida isolate that had been recovered from tilapia in the US, and they had similar cytotoxic effects to the OMB cells. So the main conclusions of our work are that we have 3 species of Edwardsia causing disease in tilapia in Brazil, and they are Edwardsia latarda, angularum, and Epicicida. E. tarda is more genetically diverse. Epicicida has more virulence genes, and E. angularum genetic groups are more virulent to OMB cells, especially sequence type 32, which is the most prevalent genetic lineage observed. With that, I would like to thank you all for your attention, and I would also like to thank my fish friends from Davis and from Belo Horizonte, and I'd like to answer your questions if you have any. Thank you.
Great presentation, Renata. That's an impressive externship project. Folks, feel free to drop questions into the chat. Otherwise, unmute yourself and you can ask directly.
Thank you. Good to see some people here.
Oh.
Kelsey.
We
I
did.
have a question, real quick,
Yeah.
Renata.
Sure.
Why
Yeah.
did you select the OMB cells in particular, rather than another tilapia cell line?
So thank you for your question, Taylor. It's good to hear from you. So this cell is from the brain of Tilapia mosambica, as I mentioned. And the brain is an important target organ for the Edwardsiellas, especially when they reach the systemic they reach systemic infection. They cause septicemia. They get to the brain of the fish. So we thought that this could be a good model to assess virulence before going to the in vivo trials that we are running right now here in Brazil.
Perfect. Thank you.
Thank you.
Miss you.
All right.
Me too.
All right. Thanks a lot, Renata.